dicentric. Okay. So what was the thing that you did the inversion, the crossing over? That part is there, but what is the output? We need to see the output. You need to show the dicentric pitch that this is the output of the crossing over. After crossing over, you will get this kind of things, and here you will find the dicentric pitch. Okay, complete that information every time. And notch one I will discuss a little bit later because next day so I will discuss that one. Okay. Uh, your quiz is going on, so complete the quiz. And next day there is first day I will give you the homework. Your homework will be this time. I will give you two experiments. The experiments are Avery's experiments and Griffith's experiment. That one, those two. The condition is that you need to show that DNA is this is the experiment. It is showing these things, and from here we concluded this part, and this is another experiment. From here we concluded this part. Okay. You need not to draw the mouse and everything, but you can do the word diagram. So the mice injected with this one, mice injected with this one, just word diagram. That is easier to understand. Okay, rather than writing whole thing, whole experiment. Okay, so we were here and we talked about this one, Mason-Stahl experiment. And this from this experiment, we knew that DNA replication is in a semi-conservative manner. Okay. Can I explain me that why these two bands are in these two positions? This is the 14N. We could get this band here and the 15N, this band here. Can you tell me why? Why not in the opposite direction? And actually, what did they do with this DNA? They incorporated the phosphorus into the DNA, leveled phosphorus, that is either 14 level nitrogen, either 14N or 15N into the DNA, and then they span it, take out the DNA, span into the centrifuge machine, and they got this kind of bands. Can you tell me why these bands are in these two locations? That is, why not the 15N here and 14N here?
It's 15's heavier.
This is 15N.
Yeah, it's heavier than the 14. That's why it's closer.
So what does it mean? What is the actual principle of this centrifugation? If you have this concept clear, you will never forget about this position of these two bands. That's why I'm just trying to get you a clear conception about that. What is the concept of centrifugation? Probably in your biology course. Yes. Why do we need to centrifuge this one, and what is the purpose of this centrifugation? Separates different components. If I will take some cells, it will separate different kind of components there. That is different organelles, cell membrane, all those things. That's fine. So, do you know the gradient centrifugation? What is called gradient centrifugation? Gradient centrifugation means you need to spin it in one RPM. Generally, we call RPM, but actually the G is the most common force there. Suppose 1000 RPM, you will spin it. Nucleus will be deposited at the bottom, and the rest of the materials are on the top. Take the supernatant, that is called the supernatant, and that is called the pellet. The hard thing under the test tube, that is called pellet, and the solution over there, that is called the supernatant. We call it a soup. Soup and pellet, these two things. Take the soup out, spin it in 5000 RPM. you'll get another P. In such case, each and every time when you are centrifuging these things, you will get different kind of materials there. At first, you will get the nucleus, like that. This is called the gradient centrifugation. So everything is there. You are centrifuging in different steps. Then you are getting all those samples isolated. Okay, this is called gradient centrifugation, and it is more or less something like that. And the principle is that during centrifugation, you will get the heavier material at the bottom all the time, based on the force you are applying. Heavier materials on the bottom. So if the heavier materials on the bottom, that means the heavier, this is the nitrogen, 15N nitrogen is heavier than the 14-1. So you will always get this band at the bottom, 15N, and 14N band on the top. If it is a mixture of 14N and 15N, you will get the band in between these two. Clear? This part? So if you have the concept of centrifugation, then you will not forget about these bands. What is the location of these bands and why? So based on this one, we discussed about these things. There are three main concepts of DNA replication. One is the conservative manner, another is dispersive manner, and another is semi-conservative manner. In the conservative manner, the old DNA or parental DNA remains same, and new, the daughter DNA is produced from the parental DNA separately, totally separate. In the second cycle, one parental DNA will produce one daughter DNA, other daughter DNA will produce other two daughter DNA. Okay? So if these two are the lighter states, that is 14N, and if this is 15N, then we will get one 15N band and one 14N band, but the 14N band will be little bit thicker because there are three things. Okay? So this is the concept of conservative mode. If the DNA synthesis is in conservative mode, we will find two bands. This band will be a little bit thicker. Clear? If it is dispersive mode, that means that parental DNA is broken down into pieces and the new DNA is formed in between. So in such case, both the DNA, the newly formed DNA, they will have both the daughter strands and parental strands. That is, if it is mixed, you will find the band in between these two. This is a mixture of band of 14N and 15N. Generally, it will be in between these two. Based on their concentration, if their concentration is too much, you will get thicker band, but you will get only one band. So in conservative, you will get two bands. In dispersive, you will get only one band. Okay? And that band is the mixture band. In case of semi-conservative manner, so one DNA, parental DNA is separating into two strands. New strands or daughter strands are forming onto the parental DNA. So the first one is a mixture of parental DNA and daughter DNA. From there, again, two strands are separating. That means one daughter DNA strand, one parental DNA strand. The parental DNA strand is forming a new daughter DNA. The daughter DNA strand is forming another daughter DNA. Okay? So there will be two daughter DNA, pure daughter DNA, and two parental strands with daughter DNA strand. This should be the mixture if it is in a semi-conservative manner. Okay? In such case, you will find the banding pattern like this. Okay? One is the 14N band and another is the mixture of these two bands. So you will find two bands, but one is on the top, another is on the middle. Actually, these are the concept. This is not the actual, because you will not find this kind of band anytime, because DNA replication is not in that manner. So theoretically, you will not get this band and this band. These are the conceptual. If it will be like this, then we will find this one, concept. So actual experiment got that result, that those two bands, and that proves that DNA replication is semi-conservative. Okay? And this is a semi-conservative manner of DNA replication. So this is the parental DNA strand, the blue one. Parental DNA strands needs to be opened up at such places. Not the whole DNA is opening up at the same time and the new strands are forming. It is just opening up for a little bit, then the new strands are forming there, and as it is going on, the DNA strand is opening up and the new strands are forming. So it is just like a zipper of your shirt, like that. You are opening the zipper and new thing is forming there, like this. Okay. Now, based on this concept, we have a couple of terms here. One is called replicon. What is replicon? Replicon means the part of the DNA or that DNA where replication is going on. Actually, the segment of DNA where the replication is going on. That is called replicon. And each and every replication needs a particular point to start the replication. DNA has a particular point. That point is required for, what is it? So each and every DNA segment has a particular point. That point is required. That is the starting point of replication, and that is called the replication origin. Okay. Generally, they have one origin. This is the origin, starting point of replication. From here, it is opening up and the replication takes place in that way. But eukaryotic DNA is long, very long DNA, having 6 billion base pairs. And in this long DNA, if the replication goes on in such a way, that is one origin and the replication is going on, in such case, it would take about 10 days to complete the replication. But the replication is done within 7 hours. In such case, they need multiple origins of replication. Multiple origins. Okay? And, okay, let's go. These multiple origins, origins means a particular sequence of DNA which can recognize a particular protein. Those proteins can come to that part and starts the replication. That is the origin. Now there are a couple of patterns of replication. One is called theta replication. Theta replication is generally in bacteria. When they are forming this kind of loop here. And it looks like this. New DNA is forming here. This is the replication fork. It is going on. It is opening like that. And at a certain stage, it looks like this. And this is the structure of theta. Theta is a Greek word. Okay. It looks like theta. That's why it is called theta replication. And another is called rolling circle replication. What is rolling circle? This is the theta replication. So this is actual DNA. That is the origin. In that origin, it is opening and it is going on. And at finally, two circles are separating. Okay. So at this point, it looks like theta. That's why it is called theta replication. And this is actually the electron micrograph of the whole DNA. And it is showing this is the new DNA forming here, and it looks like a theta. That's why it is called theta replication. And this is called rolling circle replication. So there are two strands here. This is the origin. One strand is opening up, and that strand is going for the linear replication. Other one that is going for the circular replication. Okay. So one is linear and one is circular. And this linear one, it is again making the circular one later, but at some point it was linear. This type of replication process is called rolling circle replication. Okay. Replication bubble. So go to that picture again. So this part, that is a ring-like structure where the replication is taking place, actually new DNA is forming. It looks like a bubble-like structure. Okay, that bubble is called replication bubble. As the replication is going on more and more, then the bubble is getting bigger and bigger. Okay. as this is going on this part, so at certain point, DNA looks like this. The new DNA is forming here, and this is the original older DNA. So it likes a fork-like structure at certain part of the DNA where replication is going on. That fork-like structure is called replication fork. So there are two things. One is a replication bubble, another is a replication fork. In case of E. coli, or in case of rolling circle replication, let's go to the picture again. In this rolling circle replication, the DNA replication is taking place in this way. Okay? So this is one direction replication, only one direction. So this type of replication is called unidirectional replication. And in case of the previous picture, here, the replication is taking place in both direction. That is, it is going up to that way and it is going to that way also. Okay, so the replication bubble is getting bigger, but the replication is taking place on both sides. So this type of replication is called bidirectional replication. In case of eukaryotic chromosome, it is very, very required because these are the multiple origins. So from here, it is going in this way and that way. That is the bidirectional replication. And finally, what is happening is this is going bigger and bigger like this, and this one is going bigger and bigger like this. Finally, they will meet, and the replication is done. Because for the eukaryotic, as the eukaryotic chromosome is very, very long, that's why they need this kind of replication process. So if the replication is one direction only, that is unidirectional replication. If the replication in both directions, that is the bidirectional replication. And unidirectional, the example is rolling circle replication. Bidirectional is, that is bacterial replication and eukaryotic replication. Clear? And this is the example of what I was drawing there. So there are multiple origins, replication is going on, they are extending in both the sides, and finally they meet and the whole DNA is formed, like this. Otherwise, it would take a long time. And this is our biological process. They have this kind of things there. Now, the main thing, at first we will discuss about everything that is with the replication fork and replication everything in context with the bacterial replication. Okay, prokaryotic replication. Because this is easy to understand. Then we will go for the eukaryotic replication, more or less similar, but there are a couple of proteins and other things are extra. So for the replication, we need a couple of things. Suppose you are trying to cook something. You need the ingredients first. The spices, the lower ingredients, vegetables and other things that is required for you. And you need a pot where you need to cook. You need some kind of oven, heat. This is the same kind of things. You need a DNA template first. That is called the DNA template because it is semi-conservative. So from each and every one strand of DNA, another new strand is formed. So you need a DNA template. That is the first thing. You require a DNA template. And then you need raw materials. What are the raw materials? What you need to add in the DNA? What is the component of DNA? The component of DNA is phosphate, nucleotide base, and deoxyribose sugar. These are the components of a particular nucleotide. So you need this nucleotide in a free form. Okay? So you need all deoxyribose nucleotides, that is called DNTPs. N means anything, that is DGTP, DCTP, DTTP, or DATP. Deoxyribose adenosine triphosphate, deoxyribose guanosine triphosphate, like this. Cytosine triphosphate, thymidine diphosphate. In the triphosphate form. In our cell, all these materials are available as triphosphate form, freely available in the cytoplasm. And what is the source for that? As we are eating food, we are getting those things in our body. and they are easily available as triphosphate form. ATP is very, very required for the production of energy. So they are available at cytoplasmic level. Then you need something to cook. That is your oven, you need spatula to mix it. Those are the things, think about that. They are not actually cooking. They are used for cooking only, but they are not the materials in your food. Okay? Those are enzymes which are helping to produce the DNA, but they are not the part of DNA. Those are the enzymes, okay? Like our spatula. That is not the part of our food, but you need it to cook. These enzymes are different kinds of enzymes: DNA polymerase, zydase, helicase, primase, and ligase. These are the main enzymes here in DNA replication. DNA polymerase, there are a lot of DNA polymerases, and particularly in case of prokaryotic DNA, there are mainly three DNA polymerases. Actually, five DNA polymerases are there. They are called DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, 4, and 5. DNA polymerase 3 is the main enzyme to do this DNA replication, DNA polymerization, and DNA polymerase 1 is responsible for proofreading. If there is a mismatch, they can proofread that. Why they are called polymerase? Because this is a polymerization reaction. What is polymerization? Polymer and monomer. What is monomer? What is polymer? Poly is mother. Mono means one. What is monomer? What is polymer? Try to find something. The name indicates everything. Monomer means a single unit. Polymer means multiple units. Multiple of that single unit. If I come in context in like this way, multiple of that single unit. Multiple of. So that is the polymer of glucose. Glucose is a monomer, that is the polymer. One amino acid. You made a long chain of amino acids. Same amino acid, different kinds maybe, but 20 amino acids you can make a long chain. That is a protein. So protein is a polymer of amino acids. Nucleotides. There are only 4 nucleotides are there. You are adding only the nucleotides each and every time. Okay, the same thing. Same nucleotides, 4 nucleotides, different types of nucleotides are there. You are adding each and every time. Same unit. Nucleotides are monomer. When they are forming the whole DNA or RNA, that is the polymer of nucleotide. Okay. So in the biological system, there are 3 polymer things. One is carbohydrate, one is protein, and another is DNA. Is fat a polymer or not? Fat or lipid is a polymer or not? Why? What are the multiple reasons? Fat is made up of fatty acid and glycerol, and they are not the single unit together. 1 glycerol and 3 fatty acid molecules. They are there. So actually, fat is not a polymer. Carbohydrate, protein, and nucleic acid, they are the polymers that is having the same monomers repeating. But in case of fat, that is not a polymer. So as this is, adding nucleotide each and every time, that is called the polymerization reaction. As DNA polymerase doing that reaction, that's why it's called DNA polymerase. So doing the polymerization reaction. Okay. And in case of prokaryote, we have 5 types of DNA polymerases: 1, 2, 3, 4, 5. In case we knew our list 5. Right now there are a couple of more. I will show you the list, and that is called alpha, beta, gamma, delta, and sigma. Okay. Gyrase. Gyrase is called topoisomerase. Topoisomerase means that is something. If you coil a thread each and every time, it will be overcoiled sometimes. If you coil or if you unwind a coil, suppose take a thread like this, and if you need to open this part more, then it will create tension to this part. Those are becoming shorter and shorter, it will create tension. And sometimes you will find, if you can use a rubber band and fold it several times, then you will see the rubber band is folding by itself somewhere. So it is twisting by itself because there is a lot of tension there. In DNA, that is like a rubber band, something like that. If you uncoil that part, it will be twisted in some other part. So these gyres, what they are doing, they are uncoiled those parts. The twisted parts, they are just breaking those strands and they are rejoining those strands. So they are relieving tension of the other part at the DNA during replication. That is the function of topoisomerase, and one example of topoisomerase is sidase. Okay, I'll talk about these things later. And couple of proteins, when the strand is opening up like this, they have the hydrogen bonds here. Okay, each and every nucleotide is coiled by hydrogen bonds. So they cannot remain open for a long time because hydrogen bonds will attract another nucleotide to attract them. Okay, so someone needs to keep it open. Keep it open like this. They are holding that. There are some proteins here. These are called SSP or single strand binding protein. They are binding to this single strand until the replication is going on. If the replication is not there, they are working like that. They are holding those single strand not to rejoin again. Those are called single strand binding proteins. And another protein is there that is called a beta clamp. Okay. And according to DNA structure, we know that there is 3' portion, there is OH group, and in the 5' portion, there is phosphate group. This 5' phosphate needs to join with 3' OH. Okay, this is the unidirectional process. That's why DNA strands are like this, anti-parallel. If this is in this way, the other thing is that way. Anti-parallel strand, because only one OH group, only into the OH group the phosphate group will add. If the phosphate group is open, then OH group will not add. So we need an open OH group to start the synthesis. DNA does not create that kind of open OH group, and there is no such open OH group there in DNA. That's why we need a short segment of RNA to start the synthesis. That short segment of RNA is called RNA primer. So at first, a short segment of RNA is formed here to this part, then the DNA synthesis starts. That is called RNA primer. And this is the example that OH group. For each OH group, this OH group is binding with this phosphate group. As it is triphosphate, they are having 3 phosphates. Right now, when we need to do the DNA sequencing, we can tag these 2 phosphates and they will give the light. Based on that light, we know what kind of nucleotide is adding here. Okay. So these 2 phosphates will be released. One phosphate will add to the OH group at the 3' region, and this is the 5' region. So the direction will be like this. This is in this way, and that is in that way. So the whole replication, we can divide into 3 steps. At first is the initiation, then the elongation, and termination. Initiation means the formation of replication fork, okay, and assemble of replisome. What is replisome? Replisome is the total machinery for the DNA replication. That is called replisome. Elongation means as it is initiated, the DNA replication fork is formed, then they will add the DNA nucleotides here and they will elongate until finally reach to the destination. That is the elongation part. What is termination? At the end of replication. In case of bacterial replication, there should be certain parts there where it should end, or it need to finish up to the end of the DNA chain. But the problem is that in eukaryotic DNA, the end part of the DNA contains repetitive sequence, lot of repetitive sequence. That is called the telomere. And there is some problem in the replication at the telomeric region. So that is our termination process. In the termination part, we'll discuss about these two things. So origin of replication is that point where DNA replication starts. Generally, E. coli replication begins from a fixed region and its locus is called ODC. So for this E. coli, there is only one location that is called the ODC region. That is a particular sequence there, and that sequence is the origin of replication. So from there, actually, replication starts. Now, DNA replication goes on. We know that only the nucleotides will be added into the open OH group. That is the 3' region. Okay, so in such case, this one, here it is the 3' region open group. So this is 3', this is 5'. The new DNA synthesis will be in that way, 5' to 3'. That means this is 3', this is 5'. The opposite sequence, anti-parallel sequence. And here it is 3', it is 5', that way. But if here it is 3', 5', then how the DNA synthesis will happen? There is no way. This is another problem here. So we have the DNA strand. This is 5' and this is 3'. Okay. So if the original DNA is there, then we will have this one is 3', this one is 5'. Okay, this is the original DNA. I opened up at the replication fork here, so it looks like this. So here, that means the anti-parallel strand, it should be 5', and this one will be 3'. Because in this part, always the 3', that is the OH group, is open. When the direction is 5' to 3', in this direction, newly synthesized DNA, the direction of newly synthesized DNA, the direction is 5' to 3'. This one is called leading strand. Leading strand. Direction, synthesis of direction is 5' to 3'. Okay? But here, this is the 5'. And this part is 3' region. So OH group is opened here. How the DNA synthesis can take place? That way. It cannot go to 3' to 5' because newly DNA synthesis will be in this region, right? But that is not possible because the direction is only 3' open. In such case, what is happening here? It starts from this part and goes to this way. So this is the 5' region, this is 3' region. It goes to this way up to this, stopped. Again, here is another synthesis, 5' to 3', stopped. Here is another synthesis from 5' to 3' and stopped. With the small segments. Then finally these two are joined. Okay? So this is a discontinuous replication. And this is called lagging strand. Okay? In the leading strand, in the lagging, the synthesis is discontinuous and actual synthesis is 5', 3' here. Here is also 5' to 3' direction in this way. Here is also 5' to 3' direction in this way. But the overall direction is this way, right? Because replication fork is opening up here again. And the overall direction is also the 5', 3' direction. Okay. So replication goes from 5' to 3'. There are two things. One is continuous. The continuous replication is on the leading strand, where the replication takes place as a stretch, as a whole. But in case of the other one, the lagging strand, the replication is discontinuous with the formation of short stretches of DNA and the joining of these short stretches of DNA. These discontinuous fragments were first discovered by a Japanese scientist, Okazaki. According to his name, these fragments are called Okazaki fragments. This is Okazaki fragments. Now, if you think about this one, the first thing is that in case of leading strand, there is the formation of DNA and there is no problem at all. But in the lagging strand, there are the formation of short stretches of DNA. Then the short stretches need to be joined. All this. And that is the function of ligase. Ligase can join those short stretches. Okay. But in case of actually the direction is always same, the 3', 5' region. But in case of the different rolling circle model or other things, they have different kind of patterns, but the direction is always same. That is the 5' and 3' new DNA synthesis. Now the initiation part. I will suggest you, and later probably I will give you the homework about this one. Formation of the replication fork. This is very, very important because elongation is very simple and termination is also more or less simple. But this first one, that is the formation of a replication fork or the initiation, there are a lot of things. Okay. So do it again. So this is our DNA. This is 3' region. This is 5' region. There are bonds here, like this. Okay. And the nucleotides are there. These are opened up. Like this. Okay, the first thing is that unwinding of the DNA. At first, DNA was not like this. DNA was like this. Open. That part, that part is called the origin. Okay, this is the point. From here, the DNA needs to open up. Okay, that is called the origin. Now, someone needs to determine what is the part of the origin. Which one is the origin? Someone needs to recognize. And that is called DNA A protein. DNA A protein finds out that particular ODC location. Okay, this is the ODC location. Then it can bind with that ODC location and then it can start the DNA synthesis there. Okay. So the first step is the identification of origin. What doing there? ODC, that is the sequence of the origin sequence, and DNA A protein, which can recognize that origin. Next is opening up the strands, just like a zipper. Opening up the strands. So at first it was like that, then open this up. Then open this up. Like this. A zipper, what is the function of a zipper? That function is done by DNA helicase. Helicase, the function of helicase is opening up, breaking up these hydrogen bonds between these nucleotides and opening up this thing. So the helicase is here. Okay, this is the helicase protein, which is breaking up those hydrogen bonds and opening up the DNA strands. Okay, these are open right now at this point, but they cannot remain open for long time. Someone needs to keep it open up. Okay. So some proteins, they are attaching to the strands, both sides. These are called single strand binding proteins. They are keeping those things open. Okay. These are called single strand binding proteins. Based on this opening, there is some kind of tension in the further end of DNA, a little bit far, not near here. Those part needs to be uncoiled and they needs to be relieved. The tension needs to be relieved there. That function is done by DNA gyrase or topoisomerase. So topoisomerase is not functioning immediately here, a little bit further here, where topoisomerase is functioning, little bit further, where there is some kind of tension, it will open up that part and rejoin that part. So the tension is relieved. That is the function of DNA gyrase. And then the replication fork is formed. So at this point, when helicase is opening up these DNA strands by breaking down these DNA hydrogen bonds between two nucleotides, the fork is opening up. Single strand binding proteins, they are keeping that thing open. So up to this, it is fine. But the next thing is that primase enzyme, we need an RNA primer because for the new nucleotide, new DNA sequence, we need an open OH group. There is no way DNA polymerase can create open OH group here. If there is no open OH group, DNA polymerase cannot function. Okay, here is nothing. At this point, DNA polymerase cannot function. There is no. So then another enzyme called primase. Primase. It is coming up here and forming a. So if it is A, here will be U, it is RNA. If it is G, here will be C. If it is T, here will be A. This primase is creating this short primer. Just 10, 12 sequences, that is enough. So short primer is formed and then the function of primase is done. This is called RNA primer created by RNA polymerase. This is one kind of RNA polymerase. The enzyme is called primase. And the function of primase is done. When the primer is produced here, then DNA polymerase can add here. This is in case of prokaryote, it is called polymerase 3, DNA pol 3. DNA pol 3 can add here, and from there it can synthesize all the bases according to this. Okay. Up to this, up to the formation of RNA primer, that is the initiation. Okay. Then the elongation process starts. What happened to the lagging strand? That is another thing. So here the DNA replication is at different stretches. So what is the function? RNA primase first form one RNA primer here, then his function is done. It goes to the another part and form another RNA primer here. Then it goes to the another part, form another RNA primer here, like this. Okay. DNA polymerase comes here and forms the DNA strand here, new DNA strand. Another DNA, when it is done, when this RNA primer is done, then that DNA polymerase goes there and form another DNA. Okay. What is the formation of DNA? What does it mean? It means that they are adding the nucleotides, just opposite complementary nucleotides. If there is A, it will add one T if it is deoxyribonucleotide. If it is DNA, it will add T. If it is RNA, it will add U. Because there is no thymine in RNA, uracil is there. Okay. So each and every nucleotide is added here. So it needs to code that what is there in the prime, that is on main strand. If it is there, A is there, then it will add T. If it is C there, it will add G. If it is T there, it will add A, like this. And all these nucleotides are available in the cytoplasm. It just needs to call. Okay, I have the T here. A needs to come here. Join this together. I have the G here. C needs to come here. Come here, join it. Then these two are joined by phosphodiester bond, and the new DNA is formed. Okay. So up to the formation of RNA primer, the initiation is done. And this is the way how the DNA, that is the protein, can recognize the ODC region. DNA is there and it is binding with the ODC part, that is the origin part. After binding that one, it will open up a little bit, stretch the replication fork. From here, DNA synthesis helicase can come here. Helicase can open up this fork. Single strand binding proteins are always there. They are keeping this open. And that is the function of DNA. A is done. When the initiation starts here, DNA replication starts here, it will goes off. Because this part needs to be replicated again. Okay. So this is the function of DNA A, that is origin recognition protein. To find out the origin, at first it comes to the ODC location, DNA A, binds with the ODC location, and then opens up a little space there, uncoils a certain part of the DNA. When it is uncoiled, a little part, helicase comes here, and helicase opens up the process in bidirectional way. Single strand binding proteins are attached to the single strands here, keeping it open. Then the function of this protein is done. Okay. So with the origin, this starts this part, the helicase. Helicase is going on this way, and in the later, little further, topoisomerase is there. It is just uncoiling that DNA tension there. Okay, this is the function of these three enzymes. So helicase is an enzyme that breaks hydrogen bond, just functions like a zipper. That's why I put the zipper's figure there. You can remember that one, just the function of the zipper. When the zipper is the helicase, actually, it is, if you can pull up the zipper, it will open up the strands like this. Okay. Topoisomerase, it is relieving the tension outside the replication region, and single strand binding protein, they are just keeping those two duplex open up. Now all this machinery, this whole thing together, that is the two DNA polymerases into two strands, and either two helicase or one helicase on the opening up, topoisomerase is there, and ligase is there for the Okazaki fragments. When the Okazaki fragments are formed, ligase are joining those two things, and DNA polymerase is finding here, single strand binding proteins are attaching here. All these things together is called replisome. This is the total machinery. This is the total machinery. When the DNA is formed, this whole thing is moving on to the end of the DNA. From the origin to the end of the DNA, this whole machinery is moving. That's why it is called the replisome. So what is replisome? Replisome means this whole thing, all these proteins, and there is another protein is there, because the DNA polymerase cannot attach to the DNA strand by itself. Another protein is there, that is a part of the DNA polymerase, that is called the beta clamp. Part of the DNA polymerase, because I will not go for the details of DNA polymerase structure here. It is not required, but that is a part of DNA polymerase, and that is called the beta clamp. The function of beta clamp is that just attaching the DNA polymerase onto the strand. Okay, then the DNA polymerase is called the nucleotide. Okay, I have this one, come the complementary nucleotide here. And as this one is going on onto the one side, DNA polymerase cannot act on that way. So actual the three-dimensional structure is like this. The lagging strand is coiled in that way. So the synthesis can take place in one direction. And this is the function of DNA zydase. So when there is the more tension, DNA can coils up like this. Okay, it can just release this part, two strands, just cut those two strands, keep this open and rejoin it. That is the function. So it becomes linear again. That is the function of DNA zydase. And this is happening. This is the actual picture of E. coli DNA or bacterial DNA that the coils are formed. Okay. Now, what is RNA primer? Because DNA polymerase cannot initiate DNA synthesis if there is no such open OH group. So the function of RNA primer is to supply that open OH group. And it is created 10 to 12 nucleotides long and formed by the enzyme primase. And primase is a kind of RNA polymerase. Sometimes in some book you can find the RNA polymerase as the name as RNA polymerase rather than primase. Okay. And when they can form this short stretch of DNA, the primase is removed from here and the DNA synthesis starts from that point. But what is happening to that RNA primer? If the RNA primer is there all the time, so in the new DNA strand, we will find that RNA primer and the DNA as well, right? So these are the things that new RNA primers are forming. This is the lagging strand. So in case of lagging strand, this RNA primer is formed, then another RNA primer, DNA is formed in this way, and again another RNA primer, DNA is formed this way. And in case of leading strand, it is continuous. If this is origin, so this is leading, this is lagging. If this is origin, this is leading, that is lagging strand. Because direction is different here. Okay. So what is the fate of these RNA primers? Now, with the elongation means just polymerization of nucleotides based on their strands. Template strand. What is there in the template strand? DNA polymerase reads that code and just add the opposite complementary base pair to that sequence. Okay. This is called the DNA polymerization, and DNA polymerase 3 is doing this thing. Okay. Now, DNA polymerase, these are called the polymerization enzyme. They have two different kind of activities. One is called the polymerase activity, another is called nuclease activity. So there are two terms here. One is called polymerase activity and one is called nuclease activity. Polymerase activity means polymerization of nucleotide. That is called the polymerase activity. What they are doing there? Just adding the nucleotides one by one, that is the polymerization activity. Nuclease activity means they can cut the DNA strand. They can cut the nucleotide. That is called the nuclease activity. And this nuclease activity is of two types. One is called endonuclease activity. Endonuclease. Another is called exonuclease activity. Okay. They have the specific pattern that some enzyme can have 3' 5' endonuclease activity, some enzyme can have 5' 3' exonuclease activity, like that. What is the difference between endonuclease and exonuclease activity? Nuclease activity means cutting the nucleotides. Polymerase activity means adding the nucleotides. So what is the difference between exonuclease and endonuclease? Both are the cutting of the nucleotides. But if they can cut the nucleotides at the end position of a segment, that is called exonuclease activity. if they can cut anywhere in the segment, anywhere in between, anywhere in the segment, that is called the endonuclease activity. Each and every enzyme does not have that kind of endonuclease activity. Okay. DNA polymerase 3 can have the exonuclease activity and the polymerase activity, but they don't have the endonuclease activity. So what is happening here? DNA polymerase 1 has that kind of endonuclease activity. Okay. So when the DNA is formed here with the DNA polymerase and it is getting longer and longer, just after adding couple of nucleotides here, DNA polymerase 1, that is called Pol 1. This Pol 1 comes here, cuts this part, okay, between DNA and RNA, that's part. Then remove these RNA primers from here. DNA polymerase can synthesize from 5' to 3' direction, but DNA polymerase 1 can synthesize the opposite way, 5' 3' direction or 3' 5', but in a short stretch. Okay. So what they are doing, DNA polymerase 1, removing this part with the exonuclease activity because that is the end part of the DNA. So removing that RNA from the end part of the DNA, that is called the exonuclease activity of DNA polymerase 1. Then they can synthesize this short part with the DNA. Okay. Now, all the nucleotides, complementary nucleotide sequences are there, it is formed, but only this gap between two DNA strands remains there. That is the gap between a phosphate and OH group. Only that part remains there. That part, that is called the ligation process. That joining is done by ligase. That is the ligation of DNA nucleotides. Okay. So RNA primer is removed by DNA polymerase 1. Synthesis of short segment of DNA instead of the RNA primer. That is why the DNA polymerase 1. Then there is a gap between the new DNA and old DNA. That gap is sealed by ligase. This is the function of all the enzymes. Okay. And this is, we already talked about this one. In case of lagging strand, the synthesis is discontinuous. So RNA primer is produced in a discontinuous pattern. Short stretches of DNA is produced here. Then what is happening here? DNA polymerase 1 comes here after the formation of these DNA strands, remove these RNA primers and synthesis this part. There are two joins here, this one and this one. Those are held by ligase. So our new DNA is formed continuous. Okay. In the next step, DNA polymerase 1 will remove this RNA primer, synthesis this part only, and this join is sealed by ligase. So the whole function is like this: removal of RNA primer and synthesis of short fragment of DNA. That is the function of DNA polymerase 1. And those two new DNAs are attached by just the sealing of the gap. And chemically, we can find that is the bond between these two things. That is the phosphate group and OH group. That is sealed by ligase. So these are the things. That is, here is the mRNA. That RNA is cut out from their RNA primer, and the ligation is done by ligase. And here is the main part, that is the chemical part, how the DNA ligase work there. And this is the thing, that they are joining just the strands, no nucleotide, no addition of nucleotide, just the addition of two strands. Now there are 5 DNA polymerases in E. coli. 1, 2, 3, 4, 5. We can determine these polymerases as these numbers. And functions are that there are three types of functions. One is 5 prime, 3 prime polymerase activity, 3 prime to 5 prime exonuclease activity, and 5 prime to 3 prime exonuclease activity. In both way, exonuclease activity. DNA polymerase 1 has both all. They have the polymerase activity and both directional exonuclease activity. DNA polymerase 2 has these two kind of things, but they don't have 5 prime, 3 prime exonuclease activity. DNA polymerase 3 is having the same thing. They don't have this 5 prime, 3 prime exonuclease activity. That's why that's RNA primer, removal of RNA primer, that cannot be done by DNA polymerase 3. DNA polymerase 1 is required here because they have this exonuclease activity. Clear? And these are other tools, DNA polymerases, but we really don't know the function of these two DNA polymerases. Generally, they are doing a short stretch of DNA synthesis. When we will go for the mutation part, and there we will find the short stretch of DNA, only a nucleotide is there, creating some kind of problem, or the mutation is there on the nucleotide, and DNA needs to be repaired into that part. So they can just remove that nucleotide or couple of nucleotides there, and these enzymes then they function there. Okay, so these are all characteristics of DNA polymerases. There, other enzymes, initiator protein, DNA helicase, single strand binding protein, DNA zydase, DNA primase. DNA primase sometimes is called RNA primase or simply primase. This one, DNA polymerase 3, DNA polymerase 1, and DNA ligase. These are the whole enzyme system, and that is the component of replicon. Okay, the function of DNA polymerase 3 is that it can synthesize a long stretch of DNA. Long stretch of DNA. The whole DNA it can synthesize, the whole DNA. Okay, but DNA polymerase 1 cannot synthesize that much. So DNA polymerase 3 is the main enzyme for DNA polymerization in E. coli. Now the condition is that we know that the rate of synthesis is 1000 base pairs per second. 1000 base pairs per second. This is the rate of synthesis. And in such case, there is a chance that there are some mismatch pairing. What are those mismatch pairing? It can be something like, suppose there is C here, G needs to be added, one A is added here. Okay. Can it be done? Because they have the chemical properties in such a way that they cannot do these kind of things. Okay, because they are very much that is concerned about their bonding. A can bind with T and C can bind with G only, because they are pairing, their bonding pattern is something like that. But it can be happened sometimes because of formation of two different chemical structures of nucleotides. One is called the keto form. The keto form is common. That is the main form of DNA nucleotides, and that is the common. But in rare case, if they can form the enol form or imino form, the two bond structures like these kind of things, that cytosine can bind with adenine if they have this kind of other structures in all form. They can bind with adenine because A cannot bind with C anyway, but C is bind with A in such case. Or thymine can bind with guanine in such case if they have the imino form or enol form. It can happen sometimes. It can happen, and that is rare. That is very, very rare. These kind of things are called tautomers. They are called tautomers. That is the isomers of the nucleotides which can form mismatch pairing with other bases. Those are called tautomers. And this whole thing is called tautomerization. Okay. So what will happen if there is a mismatch pair? What is the impact of mismatch pair? DNA cannot synthesize properly. It cannot produce the mRNA and other sequences. It cannot goes on. Okay. So this mismatch pairing needs to be removed anyway. And this whole thing is called fidelity of DNA replication. What is the fidelity? That means accuracy of DNA replication. DNA needs to be very, very accurate. The same DNA needs to be produced from the parental DNA. Okay? And these accuracy is one overall less than one error per 10 to the power 10 nucleotides. Like that. 10 to the power 10 nucleotides, that means 1 million. 1 million or more than 1 million, 10 million. In 10 million, there is only one error. This is the fidelity of DNA replication. How it can be possible? Because when the DNA polymerase 1 is synthesizing this DNA polymerase 3 is synthesizing the DNA polymerase 1 is behind that. Okay. After removing the RNA primer and adding this strand, it can check, it can proofread whether DNA polymerase 3 did the correct work or not. Okay. He is the guide here. He will check. Okay, if the binding is correct, then it is fine. If there is a mismatch repair, it has that endonuclease activity, it can cut from the middle, it can cut out that part because all the enzyme and nuclease activities it has. So DNA polymerase 1 cut out that mismatch part, synthesize or add the actual nucleotide there, and then ligase can join that part. So this is called the proofreading. DNA polymerase 1 is the responsible enzyme for this proofreading. It can proofread the function of DNA polymerase 3. If there is mismatch pairing, it can remove that pair and can replace that nucleotide with a new nucleotide 1. So the DNA replication is more or less perfect. Definitely it should be perfect. It is always 100% perfect. Otherwise, we will be different organisms very soon. Okay, now the part is that termination. So bacterial DNA is circular. So how much it can go, that the replication, it should be terminated in a particular space. And there are different, that is, observation, different research is going on with this termination part. And you can find a little bit about this one in your book, but this is mainly from this book. That is the Levin's gene, Benjamin Levin's. Probably Levin is not alive right now, so Krebs are doing these things. Benjamin Levin's gene, that is a very good book for this one, and Watson's Molecular Biology of the Gene. These two books are really very, very good for DNA replication. And another book is there that is called Cooper's Genetics. Okay, these three books are really good. So in case of E. coli, there are some sites. Those sites are called termination sites, and they are overlapping sites. The function of those termination sites or TAR sites are that they can block the replication at that space. Suppose if the replication is blocked in the K phase, so it is going on in this way, one, and another is going on in that way. So if it is blocked in K phase, case point, will J work to block the DNA again here? In such case, there will be a gap. One is stopped at K and another stopped at J. Okay, this part is not been synthesized. The very interesting fact is that though they have a lot of termination sites, but if one termination site is active, other termination sites are not active at that time. So if K is active here, then all of the sites are not active that time. So that means this part will stop at K, other part will go up to the K to stop the termination. But they have different sites. These are called TAR sites. And another thing is that there is another protein is there that is called the TAS protein. Actually, TAS protein determines where to stop. TAS protein comes here and binds with any one of the TAR sites. These are all the TAR sites. So task protein comes here, activate one TAR site, and then that task TAR complex, that will give the signal that this is the termination point. Though there are a lot of TAR sites, but when the task protein comes here and activates one TAR site, then they can signal that, okay, this is the termination point of DNA replication. So other TAR sites are not active at that time. And this is in case of bacterial DNA. So there are two things. One is TAR site, and the function of TAR site is by task protein, that is called the task TAR complex. Okay, I think I'll stop here at this point because eukaryotic DNA is little bit complicated, little bit, not too much, because there are histone proteins. In bacterial DNA, there is no histone. But in eukaryotic DNA, the histone proteins needs to be replicated. Otherwise, you have in one DNA there is histone protein, but in other DNA, if there will be no histone protein, then what will happen? It should be identical and similar, both the DNAs, the parental DNA and the daughter DNA. So if there is histone protein, that histone protein needs to be replicated into the new DNA. And the new DNA is formed. So there are two processes there. That is one function. And the second function is that eukaryotic DNA is very long. So they will create a lot of bubbles, replication bubbles there. And that is bidirectional. That is second point. Third point is that the termination point. Because termination point there is called the telomerase region. Telomere is available in all the chromosomes, but telomerase is not present in all the chromosomes, all the cells. Okay, telomerase is available in the gamete cells because gamete chromosomes need not to be disturbed anyway. Telomerase is there. Brain cells and some other cells, they contain telomerase, but all the somatic cells doesn't contain telomerase. Okay, these somatic cells where the telomerase is not there, then there is a chance of shortening of chromosomes. Okay, so these 3 points I will discuss next day. Then I will finish this eukaryotic replication part and I will start the transcription part. These 3 classes, that is DNA replication, transcription, and translation, I will just join each other with 1 to 1. Okay, and these are there are a lot of things here, and you need to learn it very well. If you learn it one time, because I have the map, the whole map, try to learn it with a picture that is going on. Next day I will show you the some of the videos that how it is. But I have 5 minutes. I can show you one video today. And try to remember the picture always.
Initiation of replication requires a particular DNA sequence called the origin. This sequence is called ORIC in the E. coli chromosome. A protein called the initiator protein binds to ORIC and causes the DNA to unwind. Unwinding requires several proteins. The most crucial are DNA helicase, which breaks the hydrogen bonds that hold the 2 DNA strands together. Single-strand binding proteins, which help maintain the DNA in the single-stranded state. And DNA gyrase, which reduces torsional strain. An enzyme called primase synthesizes short RNA primers to allow DNA synthesis to begin. On the last RNA nucleotide of the primer. Because the 2 nucleotide strands of DNA are anti-parallel, replication takes place continuously on one strand, the leading strand, and discontinuously on the other, the lagging strand. The short lengths of DNA produced by discontinuous replication of the lagging strand are called Okazaki fragments. DNA polymerase I cuts out the RNA primer and uses 5' to 3' polymerase activity to replace the RNA nucleotides with DNA nucleotides. DNA ligase seals the nick between the DNA added by DNA polymerase III and that added by DNA polymerase I without adding another nucleotide to the strand. Through these steps, the replication machinery of the cell uses the parental DNA as a template to synthesize the leading and lagging strands.
Okay. This is for today. Read it carefully. Already I posted the slides there. So you have to have a very good idea about this. Because this is the base of all biotechnology. Later, when we'll talk about the DNA sequencing and the PCR reaction, everything is based on these three parts: replication, transcription, and translation.